like to work hard to listen. I want to like think hard about the science. But this is interesting, and there is a non-zero chance that I'm going to probably break out into karaoke at some point because I'm ready for that. All right, so I'm Angie Laird. I'm at Florida International University in Miami. I have been doing meta-analysis for almost 20 years now. And so I have a lot that we can be chatting about that. For me, the emphasis as we're thinking about meta-analysis is that it is actually about reproducibility. So a brief historical perspective, because as you advance in your career, you get to the point where you can look back and you can see how these trends evolve over a number of years. And so before this sort of current and ongoing reproducibility crisis, there were a number of investigators who, since the very dawn of let's put people on the scanner and map and see what's going on in their brains, there were people that were interested in the amount of variability that you get with any particular group of participants or any particular task. And so when I started learning meta-analysis in the early 2000s, there were a couple of papers that were out there from the mid-90s that's what I learned about meta-analysis from. And so there was a lot of work by Thomas Paus and then the Picard and Strick paper where they were looking at variability of activation patterns across the cingulate. And you know, these days it's like every year there are several papers that come out that look at meta-analyses of medial prefrontal areas. This is just something that since way back in 1996 and even a little bit before, people are interested in the variability that you get along the medial cortex. So there are a number of papers that showed us from the very early days that there's a lot of variability in across tasks, across participants, across methodologies. And one thing that from the very get-go has been really crucial is that our field is based on two very particular community standards. One is that we take individual participant brains and we spatially normalize them to a group template so that we're all talking about the same space approximately. And once we have a nice blobby map, we extract the centers of mass of our clusters as XYZ coordinates onto that standard template. So while the field itself is all about, hey, let's publish a paper and put a figure of a really pretty group brain activation map out there, that's just a two-dimensional representation of a set number of slices in a nice glossy journal page of a really complex three-dimensional map. And the coordinates that get extracted for that, that's sort of our gross approximation where we can give you a quantitative measure of where this thing happened in the brain. And so yes, you lose the three-dimensional extent and clusters shape, but you still get that center of mass that can be easily extracted from papers. Those XYZ values, I don't have to have your statistical parametric map, I don't have to have your raw data, I have your coordinates, and that gives me something that I can do meta-analyses on. So there were some early meta-analysis methods. One was the Fox-Lancaster paper in 1997 of functional volumes modeling, where they tried to come up with a statistical technique to take those XYZ coordinates and look for consensus. That one didn't really catch on. There was, in this era, a really interesting paper by Gordon Schulman. It was a two-part paper that was common blood flow changes across different tasks. The first paper was increases in cerebral cortex, and the second paper that doesn't get talked about very much was decreases in cerebral cortex. And that was the paper where they were like, we're looking at these decreases across these different tasks and the same regions pop up and we don't know what's going on with that. And then there was, of course, the 2000 paper that was kind of subsequent to that, where of course Marcus Raichle told us all about, hey, default mode brain network. So again, it was sort of this meta-analytic approach that kind of led to that. So from those very early days, meta-analysis has been really important in highlighting really important things in our community, the default mode network being one of them. After that, there were a couple of papers in the late 90s and early 2000s, one being maybe we should have a repository of these coordinates and establishing the Brainmap database for facilitating meta-analysis by creating an archive of the coordinates. And the second one there on the right, the Turkletop paper in 2002 that introduced the activation likelihood estimation paper. So for me, these papers really sort of form how the very early days of meta-analysis evolved into what our current state of the art is. So let's talk about let's back up a little bit and talk about why we need meta-analysis in the first place. We know that there are limitations to our studies. One is that we have relatively small sample sizes, particularly compared to other fields of cognitive and social science, and as well to clinical research. We also are dealing with the fact that in the case of either BOLD, fMRI BOLD, or PET, we're looking at indirect measures of neural activity, and so our reliability is limited by biological, technological, methodological confounds. In addition, our literature is just chock full of isolated findings. We know that novelty and innovation are rewarded in our high-impact journals. Historically, we're not really very good at replication and reproducing other people's results, so the literature is very much full of do this logistics and expense. Looking at confirmation, looking at extension, that's just really rare. So that increases the variability in our literature. In addition, we have a lack of focus on generalization of context-specific findings. And so the inference of what we can say about brain function and pathomechanisms, it can be limited based on what the bulk of our literature focuses on. Okay, so again, I said before that we have these two things going for us, one that we do spatial normalization and one that, typically speaking, people do publish their XYZ coordinates of activation. And so what that means is if you take a big stack of papers that all kind of looks at the similar type of task or cognitive construct, you can get a nice bunch of tables that tell you where they observe their brain activation patterns. And if you just do something simple, like they did in the early days of meta-analysis, which is plot those coordinates on a standard brain template, even though that these are the centers of mass coordinates, you still see where we're finding consensus and where we're seeing a lot of variability. So when we really started thinking about, okay, it's not just a matter of plotting those coordinates, we really need to do some sort of statistical analysis to see where truly there is consensus and where there is a lot of variability. There was some methods that were developed where we're looking at primarily kind of the focus for coordinate-based meta-analysis is to convolve the foci with some sort of spatial kernel, trying to figure out what that kernel needs to be. There's been some different studies on that, but basically taking study-specific modeled activation maps, generating those from those coordinates, combining those MA maps to a sample-wise map, and then comparing them to a distribution to determine voxel-wise statistical significance. That's the general sort of framework for a coordinate-based meta-analysis. I will talk about specifically the activation likelihood estimation method, the ALE method, because that's what I've had the most experience with. So graphically, each foci is modeled by a 3D Gaussian distribution. So those XYZ coordinates that you pull from the paper, it's just a point in space, but if you convolve it with a Gaussian of some full width half max, and you do that for all of your activation points, your peaks in your study, then you can look at where there is, you can look at generating that modeled activation map. You do that for a number of studies, and you look at the null distribution to identify what is statistically significant, and you end up with a nice little map. Okay. So there are also quite a number of other coordinate-based meta-analytic approaches. I point to the Samartzidis, I'm so sorry for butchering his name, paper that kind of summarized methodologically how these different techniques differ from each other. Alternatively, there is image-based meta-analysis. And so the question to ask here is, let's say you have those statistical parametric images and you don't have to do this sort of approximation, taking your 3D peaks and blurring them up to a rough idea of what the original map is. Say you have those maps, well then there are other meta-analytic approaches that you can leverage to do a much more precision-based identification of consensus across studies. And so Camille and Tom did a nice detailed summary of those different methods out there. The thing with the image-based meta-analysis is that there aren't, at this point, a lot of freely available SPMs that are openly downloadable for the community. We do know that NeuroVault is the repository for those. Please upload your own maps. Even if you aren't interested in meta-analysis, someone who comes along behind you may be. So if we're going to move the field forward in taking us from the predominance of coordinate-based meta-analysis to image-based meta-analysis, which theoretically would be the preferred approach, then we need more data. Those coordinates are easy to pull from the PDF. You assign that to a poor undergrad, give them a stack of papers, and they have to extract those coordinates. And so that's doable, but the preferred approach would be if NeuroVault was this hugely robust repository that had so much data for us to capture. Okay. So then I wanted to say something about what can you do with meta-analyses and what are people doing with meta-analyses? And before doing that, I should say meta-analyses are getting pretty popular. And so there's different literature searches that I can do to kind of index and quantify that popularity. One is just looking at papers that cite the original AL meta-analysis method. Another one is looking at papers that cite Tao's first Neurosynth paper that proposed Neurosynth back in 2011. And you can see the number of citations is increasing over time. Another way of doing it is just looking for any paper that has a keyword of, let's say, fMRI plus meta-analysis, also increasing over time. But I wanted to say something a little bit about, well, what are people meta-analyzing? And I had this old slide where we had a list of different meta-analysis topics. And that slide was just outdated and not comprehensive, so I took some of the abstracts from the pink slide and I put it into a word cloud to try and get some idea to show you that there are a lot of different topics that people are doing meta-analyses on. This one made me laugh because, dude, my name is super big font. So I pulled out all the author name and I made you one that's a little bit better, a little bit bigger, that gives you an idea of sort of that full spectrum of, yeah, we all need a little bit of self-reinforcement that our research has value, so you get the Laird one. Anyways, so word cloud, just to give you an idea that we're really doing a lot of different meta-analytic work across the full spectrum of cognitive constructs and tasks. So the next part of this, I wanted to focus on, you're sitting in the audience and you're like, oh, meta-analysis, this is not really relevant to me. And so I would like to offer a counterpoint that there are certain things that meta-analysis can do for you even if you're not interested in meta-analysis. I start with a basic premise that identifying a consensus and figuring out where you are within your own research community and sort of where there's variability and where there's consensus, that's sort of meta always a good thing. But let's talk about some specific examples. So say you're a new trainee and you're starting a new project and your advisor, your supervisor, your mentor has said, I want you to work on project X. And you go, oh my goodness, I don't know anything about project X. I've only studied project Y. I'm not really sure what the literature looks like in this domain. And so it's a great introduction to a new topic that often results in a formal publication. So you can kind of learn about the state of art in your field while you are boosting your CD. So if you are new to a field, meta-analysis offers you an opportunity to become more expert in it without acquiring any new data of your own. Number two, and this is the big one, is your advisor says, oh, I would really like for you to mentor this undergrad in your laboratory, and you need to give them a project to work on. And you go, ooh, that's tough, because undergrads, they don't always have a long period available to complete specific projects. Sometimes you only get them for a summer because they're participating in some internship, and you need to give them something that they can actually do in a set amount of time without having to go through 2 years of training, of the level of what we're doing here. And so you say, oh, a meta-analysis. I can have them select a topic. What we like to do is we have them pull from an existing, I'm doing some, mentoring some high school students this summer, and we had them pull an existing meta-analysis, and then they had to go get the papers, they had to read them, they had to extract the coordinates, and they basically had to replicate that published meta-analysis. It lets them have an image at the end of the day. The software is very easy to use, which I'll talk about that later. And it just gives them something that they can say, this was a project. And you can feel good that you supervised the next generation, but you did it in a way that it was feasible and not a nightmare. So again, I really like meta-analysis for that particular reason. Number 3 is, okay, I'm a very experienced researcher. I have been in my field for quite a while. I know what's going on. But, you know, there's been a bunch of papers that have come out recently, and I'm really interested in sources of variability that I think are particularly important, whether or not it's some sort of group difference from a clinical population, or whether or not it is some variation in how a task is being done consistently, or a range of tasks for a particular cognitive construct. 1 and 3 are pretty similar. It's just that 1 is, I'm trying to figure things out, and 3 is, I'm ready to really show the field something very new that we're not really realizing that is an area of conflict. Also, there is a validated new task. We had a paper come out early days of meta-analysis where someone wanted to use a self-paced end-back task in a Parkinsonian population. And they were having trouble getting the people to stay with that quick, fast-paced end-back. But they wanted to show that their self-paced version was actually identifying that same sort of cognitive network as a regular end-back. So it did the meta-analysis of end-back, and then we compared it to the self-paced in that particular population. So that's a good example of how you can sort of integrate meta-analysis into a paper about the population that you're interested in. Or validate a data acquisition approach. The very first meta-analysis by Peter Turkeltaub using the ALE approach, the whole reason he did that is he was interested in single word reading using fMRI, and all of the prior literature had focused on using PET. And so he was like, well, I'm going to do this meta-analysis of all the PET single word reading literature, do my task in fMRI where I'm sort of jittering the acquisition so that I can get good maps and show that it is comparable to the PET maps, that the same sort of network is being identified using fMRI. Number 5 is that you can use meta-analysis for ROI selection. So you have this particular study where you know you are interested in social regions or emotional regions. You can use meta-analysis to help you zoom in on what those regions are and identify specific boundaries of your regions of interest that you can then go probe in your own data. And you know, there's something to say about this potentially circumventing some of the double dipping concerns. And also it's just a more robust, objective way of saying, I'm interested in this type of a region, I'm going to identify what it is and then go target it in my follow-up study. 6 is for functional decoding, and that is basically, we all want to, in our discussion sections, sort of talk about the functional significance of our results. What do these maps mean? What do these regions do? Here's this region, what is going on here? What sort of tasks is it responsible for? And meta-analysis can help with that. And then last, you can use meta-analysis as a data-driven approach to validate much more sophisticated cognitive models. And so these last 3, the ROI selection, functional decoding, and 7, these are all sort of critically important to me as thinking about what meta-analysis has to offer. So I have a couple of slides to follow up where I'm going to go through each of these in a little bit more detail. So let's start with the ROI selection. We did a meta-analysis of problem solving. Problem solving is one of those things that it can mean a lot of different things. So we basically set forth a very strict operational definition of what problem solving meant for us. And we did a meta-analysis of all of the studies that met our inclusion criteria. And we got a lovely map of a fairly straightforward, what you would expect, nice frontal parietal. Hey, we're thinking about problems and we're solving them. And the reason we did this is because we had just embarked on a longitudinal study that was looking at how people who want to be scientists start learning the scientific method. So we took undergraduate students at the beginning and ending of their first introductory physics course, and we scanned them while they were doing this force concept inventory, which is a standard behavioral pen and paper measure of physics conceptual reasoning, where you give them some physical scenarios, you ask them something about the physical scenario where they have to reason through different physical principles, and then you give them multiple choice and they answer what actually happens in the outcome of that physical scenario. And so we knew we wanted to look at regions associated with solving complex scientific problems. So we took our meta-analysis images and we asked them certain questions. It had specific hypotheses about accuracy, difficulty, and the strategy associated with problem solving and accuracy in our cohort. So we took our meta-analysis map, In the meta-analysis itself, you know, it's one thing to say, here's all the problem-solving regions and show that big global map of that, but it's also really interesting to look at the different categories of how you can break your meta-analytic cohort down and say something interesting. For us, it was about, we had specific problems associated with mathematical problem-solving, verbal problem-solving, and visual spatial problem-solving. So sort of that presentation domain was interesting for us to look at how the maps were similar and different. And so in the conjunction image below, those were regions that showed up across all 3 different representational domains. So those were the ones that we went after in our study. We also looked at some memory-related ROIs from 2 other meta-analyses that focused on declarative and autobiographical memory. And what we ended up finding was that when you looked at these particular regions and you looked at task activation and whether or not that there was modulation according to accuracy, difficulty, and strategy, we found that difficulty, but not accuracy or strategy, modulated the brain activity during the physics conceptual reasoning. And so this problem-solving network that we see there on the right-hand side, it was engaged whether or not the correct answer was achieved. So students that are thinking about physics are accessing this particular network, and it increases as the difficulty of question increases. So anyways, this was, again, just to highlight the fact that maybe you have a particular study where you have some specific hypotheses about memory and reasoning. Starting from a meta-analytic approach to help you see those ROIs can often be really helpful in a more objective, systematic way, rather than just saying, okay, I'm going to pick this region here. Okay, so functional decoding is another big one. There are 2 papers that I would direct you to by Russ, one from 2009 and the other from 2011. And this is where we've really started talking as a community. He generated a lot of conversation with these 2 about forward inference, which is, okay, if you have task activation in this particular circumstance and you observe brain activity during that task, whereas reverse inferences, given this brain activity, therefore I'm concluding that mental function Y was engaged. And this simple way to word that is, you can say fear task activates the amygdala, that's your forward inference, and the reverse inference is, well, the amygdala is activated, therefore this participant was experiencing fear. And so Russ sort of clearly walks us through these 2 papers, why the reverse inference is not such a good thing. And it's just hard to really convey how pervasive this is in nearly every single discussion section. Like, we know reverse inference is bad. We know the amygdala activated, therefore fear was engaged. We know that's bad, but when we sit down to write our papers, it's hard not to do this. It's hard to not let a little bit of this bleed into sort of our speculatory comments about what it all means. And so when you're writing your discussion, I would really encourage you to take a step back after you've written it and then look specifically for cases of reverse inference that you're making throughout the paper, and then find ways to reword or maybe do something better. I know that of all the papers that I've read in my lifetime, they've shaped my worldview and how I think about brain regions and what they do. And that's just going to be the case for all of us. We're not machines, right? But we have machines, and machines can help us with some of this problem. And so that's where we start talking about, in those 2 papers, whether or not we can use databases of coordinates that help us do automated meta-analyses, that help us with functional decoding, so that we're doing this in a much more objective manner. You know, I don't know how many papers I've read in my lifetime. It feels like infinity, but it's not. But when you're talking about Neurosynth with 11,000, 15,000 papers, you're talking more than I have read, right? And so there's an opportunity there to develop meta-analytic functional decoding approaches to allow you to make inferences about what it all means in a way that's much more objective. And so one of the first ways that we did this using the Brainmap database was in a J Neuroscience paper in 2009, where we were looking about regions of the default mode network and sort of mapping out what behavioral domains, how they varied across different nodes of the default mode network. Another really good one, again, one of these Meteor Prefronter papers, was Alejandro's more recent paper in 2016 doing this using Neurosynth. I put both of these up here primarily for the different in visualization, because this is the thing, is it's easy to do reverse inference in an informal way because you're writing text and you're just talking about the regions. To do it in a more formal, automated way using a meta-analytic database, there's data behind that, and that means you have to figure out how to visually represent this information in a way that you can actually read the font so that your people who are reading the paper can understand what's actually going on. And so if you look at decoding papers that put this in here, some people do it in tabular format, some people do these little fingerprints, some people use the bar graphs. I'm still looking for the optimal approach. I think it kind of matters on what your application is and what your results do, but that's true in any case, right? Visualization is always a big deal. Okay. So these large-scale meta-analytic databases, the whole purpose of them is to consolidate results from many studies. And at this point, I am sort of talking specifically about Brainmap and Neurosynth, and there are some differences between those. Integrating the algorithms into the databases make performing meta-analyses easier. And so specifically Brainmap and Neurosynth, they're very complementary. And so we have this little table here that kind of highlights, Brainmap was created earlier. It was the sort of first approach to saying, hey, we should be creating an archive of coordinates to facilitate meta-analysis. Neurosynth came along later with a much alternate approach. Brainmap is based on manual annotation. There are literally RAs in a cubicle in San Antonio who take a paper and extract the coordinates and extract the metadata and enter it into a piece of software called Scribe. If you think your postdoc was bad, try doing that for a couple of years. So I learned a lot about like what it is to try to extract meaningful information from papers. Neurosynth, alternatively, it's much quicker. It is going to use an algorithm to scrape coordinates from tables and automatically populate the metadata by looking at author terms that co-occur in an abstract. And so because of the manual versus automated difference, you get difference in the annotation unit. Brainmap, you have a human being reading a paper and they can say, oh, look, this is task versus control, and this is task versus rest, and this is control versus task. And all of those get input separately. Whereas with Neurosynth, it's scraped. So everything is sort of annotated at that paper level and there's no digging down of separate experiments or contrasts. But because manual annotation is so labor intensive, Brainmap is only about 3,000 papers. And Neurosynth, this was my last tally. I don't know where we are, Tal, in terms of volume, but it's much, much higher. So the idea is that you really need to think about, do I want Brainmap or do I want Neurosynth based on what you're going for? And I have a separate slide about that. Both offer tools for meta-analysis and decoding, but again, both are coordinate-based, not image-based. Okay, so I do want to get to my one last example here to kind of stimulate your creativity about how meta-analysis can be helpful to you. And this last one is our data-driven approach to validate cognitive models. Okay, so this particular avenue of research started out from the perspective of, wouldn't it be great if there was a cognitive ontology, that there was this formal way of describing what different tasks were and what different behavioral domain cognitive constructs were, and that we had this good way of annotating all of our studies. And so we have so much work that has been going on in the last 10 years for management, sharing, and meta-analysis of our neuroimaging big data. And our knowledge representation systems, they have not, I mean, in some ways, for example, BIDS, like obviously that has been very, very productive, but getting scientists to come to a consensus on what are the tasks and what behavioral domains do they link to, that's been much more challenging. We've been working on it with our CogPo, Cognitive Paradigm Ontology. Russ has been working on it with Cognitive Atlas, but there's still a lot of work to do. And so as a result, our cognitive data descriptors are relatively underdeveloped. Again, I mean, I've seen it personally. You put scientists in a room and you ask them to talk about tasks, and there's going to be a lot of argument, even one person arguing with themselves, right? I mean, this is just a complex problem to solve. So we kind of took a step back and said, you know what, maybe let's see if there's a way of looking at this in a more data-driven perspective. And let's see if we can identify consensus from that data. So the premise was, if there are differences in activation patterns within a given group of tasks, then maybe that is important information that we should be capturing with our taxonomy and our ontologies, as they indicate meaningful segregations in brain function. And that if you have similar brain networks, maybe those should be grouped as similar in terms of tasks. And so, yes, and then with our different patterns, there should be, those types of tasks should be classified as functionally distinct. And so this is where we started from in the perspective of, can we use an approach like this to help us refine our ontology? But I will say, as this has moved forward, it makes more sense to me just even as a general meta-analyst, because if you do a meta-analysis of, let's say, problem solving, and you lump all your coordinates into one big group, then what you're saying is, don't worry about co-occurrence of which sort of regions are talking to each other the most and in which context, just what nodes pop up the most. I'll clarify that a little bit more after we talk about the methodology. The whole idea is you extract your coordinates from your literature like you do in a standard meta-analysis. You create these modeled activation maps where you're basically just reapproximating those statistical parametric images, and then you generate a correlation matrix of all of those maps. You're trying to identify which maps are similar to each other. And I would argue with Pierre from yesterday, the dendrogram doesn't tell you anything. Well, if you run hierarchical clustering, this does give you a bit of a visual representation of the similarity across groups, across your clusters. And so it may not be sort of quantitatively helpful, but it is sort of visually qualitatively helpful. And so in this particular case, we're looking at 4 different clusters of activation maps that are all approximately similar to each other. We started doing this using hierarchical clustering. We have since moved on to k-means, but this does give us that dendrogram that gives you guys a representation of what was done. So the idea is you throw a bunch of maps in there and you try and group them by the ones that are similar. The particular example that I just showed was face perception tasks. And so we got 4 different clusters, and if you run a clustering analysis on your data, you got to kind of figure out what it all means, right? But with meta-analysis, we have the metadata. We know either Brainmap or Neurosynth, depending on what you use. We either have terms or we have the formal manually annotated taxonomy to tell us what type of task and what were the participants doing, what were the stimulus response instructions. And so we can actually take these clusters and then take a step back and look at the metadata that was driving these differences between groups, and we can make these inferences about what these different clusters are doing. So in this first one, the metadata told us that we were looking at visual attention and visual motor coordination to faces, and that primarily this was activating the salience network. The second one was perception and recognition of faces. We see the amygdala there, we see the fusiform face area. The third one was social processing and episodic recall of faces. We see that lovely nodes of the DMN. And then the last one looked to be, in terms of the tasks that fed into this cluster, face naming and lexical retrieval. And so what I like about this is that it offers you an opportunity to look at cognitive models within your particular domain. And so there's the Gabanian-Haxby model of face perception that was originally purchased, paywalls, in 1996, and then updated in 2002. And so we kind of looked specifically at that one and to see whether or not our results align with that particular model. And indeed, we saw quite a lot of overlap between what they called the core system, that was our perception and recognition of faces, the extended system that to us focused on that social processing and episodic recall, with a specific sub-component there that we found as that 4th clustering for the naming of faces. But what we didn't see was something that sort of captured that idea of salience. And so this became a paper for us in 2015 where we focused on confirming and extending a well-known cognitive model of face perception using this meta-analytic approach. And we were able to demonstrate that this large-scale data mining can inform the evolution of these cognitive models. Like it's very, very important for cognitive scientists to sit down and come up with cognitive models, but it is just as important that we sort of, as a community, look at our data and see whether or not we're in alignment with those so that we can nudge them along and improve them and evolve. We like this approach, so we've actually done it a couple of times recently. Katie Bottenhorn has done it in the context of naturalistic tasks, and so she was able to develop a lovely little figure there about the relationship between, instead of just calling them clusters, we started calling them meta-analytic groupings. So we've got different mags there that represent the input, visual and auditory, our attentional control, and then the language, emotion, and navigation processing. We also have done this in the context of emotional tasks. This was Cody Rydell's work, where we were able to confirm the Oxner model of emotional processing. We've also done this more recently. These papers aren't available just yet in reward tasks and looking at value prediction and value modulation. And then Julio, who is here today, is in the process. He was able to give us a sneak peek at some of his results that relate to pain tasks, where we see different flavors of the salience network. And so the fun thing for me, like the reason we hang around is because science is fun and the careers get long, and it gets to be such a joy to kind of see how these things progress and take a step back and say, well, what are we actually learning here? And so this series of papers for me really drove home this idea of large-scale brain networks, and coming from a meta-analytic approach and seeing things that keep popping up in different meta-analyses, it really shapes how you feel about brain networks. And so across these different phase perception, naturalistic, emotional, reward, and pain tasks, we are consistently seeing those three networks that all of us talk about all the time: salience, default mode, and central executive. And that's not really surprising, because we had a couple of papers that started out with Steve Smith's paper in 2009, where we did ICA of the Brainmap database and ICA of resting state data, and identified that there was significant correspondence between the different resting state networks that popped up in both. But you can't really call Brainmap ICA networks resting state networks because they're all from tasks. So this idea of this intrinsic connectivity that can be identified both at task and at rest, that part of this really shows that the salience, default mode, central executive, that they keep popping out in our domain-specific meta-analyses, it's not really anything new here. I had a follow-up paper in 2011 where I kind of delved into more specifically the different types of tasks that activate these networks. That's really useful in characterizing, like, what does salience do? What does ventral attention do? And you know, we did have this other little silly paper that came out in Ticks, where we basically took all of the Brainmap database and threw it into one big meta-analysis. And so again, it's no surprise that we see basically the salience network, specific regions, that cingulate and insula, that pop up in so many of our papers. So because of this, and because of Menin's tripartite network model, a nice paper that kind of talks about the importance of these three networks in the context of psychophysiology, for me, coming back to these sorts of papers, it really resonates with me because of all of the work that we do in meta-analyses and how often these three networks keep popping up. So when we had a study where we were looking at these undergraduate students and their experiences with STEM anxiety, we know that there are sex differences in STEM anxiety, that women and men have different experiences in scientific domains. And so looking at sort of how the brain and anxiety are associated with each other, given the amount of work that's been done in the tripartite networks with anxiety, and given what I feel, given my experience in meta-analysis and those three networks, we sort of focused specifically on those networks when we were doing that work, and that really kind of drove how we proceeded with this particular paper, which is currently in revision. So, with all of that, I'm hoping that it's clear that there are certain things that possibly meta-analysis can do for you. I think the three things to really emphasize there are the ROI selection, using it for functional decoding, and perhaps using it to validate cognitive models. So thinking about it then from a more methodological viewpoint, there are these different methods that are available to us. So there are options for you to do your own meta-analysis. And so, okay, maybe I've convinced you. What do you actually want to do next? Chris mentioned Anima. Anima is a meta-analytic database of images. So if you do a meta-analysis and you publish a meta-analysis, it would be really great if you would upload your meta-analytic images to Anima. But it's also great if you're starting out and you want to kind of browse for ideas. We do want this source to be added. But at this point, it's nice to kind of just get you thinking about what's possible. And then once you do, there are database and software available for you to do meta-analyses. If I had to summarize, I would say if you want to do a very detailed, manually annotative, paradigm-based, so you're interested in the Stroop task, or you're interested in the NBAC task, or you're interested in working memory tasks, you want to potentially consider using Brainmap to do a very detailed, heavily annotated meta-analysis. If you're looking for something where there is an option for doing automated and fast meta-analysis with excellent decoding options, then consider Neurosynth. So it really just depends on what your application is and what you're doing. I will say that both of them offer the tools for meta-analysis. Brainmap uses AL. Neurosynth offers MKDA, as well as annotation, where in Brainmap we have our structured taxonomy terms, and in Neurosynth we have our author terms. And again, those have a lot of pros and cons in themselves. Now, as I move into the next part here, I will say that currently these methods are spread out across a lot of programming languages and user interfaces. And so I have this whole thing down here where I'm saying, maybe you want to use this one, maybe you want to do this one. But wouldn't it be great if there was an opportunity that there was sort of a one-stop shop where you could go and perhaps pick and choose? We have started working on Nightmare, which is a neuroimaging meta-analysis research environment. The idea is that rather than have things spread out across different programming languages, some closed source, some open source, different interfaces, let's go ahead and develop a common interface where you can utilize these different algorithms to all work in your existing Python ecosystem. And so this is something that Taylor Sallow, a graduate student in my group, is currently working on. He delightfully encourages anyone who wants to contribute to the growing Python package to do so. We're hoping to get this released out to folks and to integrate it with some of the other tools. In fact, we have kind of a representation here of this broader Python ecosystem where you have your first order fMRI analysis tools, and then you've got here the relationship laid out with Neurosynth, Brainspell and Metacurious, the Cognitive Atlas. Just the idea here is that we want to enhance the user's experience and not send them out to go a whole bunch of different places, particularly when those other sources may be closed. So stay tuned for Nightmare. Hopefully we'll have more to report on that soon. I've got a little bit more time. One thing I noticed when we were looking at the applications for those folks interested in attending Neurohackademy is that I definitely saw a lot of folks who are interested in open and reproducible neuroscience, who maybe are a part of labs where the PI is a little resistant and a little concerned about what that actually means, and it's too revolutionary. And so I thought I would kind of summarize some things that you could maybe be thinking about at a bird's eye view of things to take home to your home institution that may be better received than your PI thinking that, oh my God, she just wants to post all her data open and we're going to get scooped and it's going to be super scary, right? It's much more than that. So we heard this morning about the data processing pipeline, and so using Git bids, getting your data formatted in bids format, and using fMRI prep, and using MRI QC, these are things that will only enhance your lab's pipelines, regardless of how open they are. If you are doing them internally, this is something that you can convince your PI of, like, look, I'm going to graduate, or I'm going to move on to the next phase of my career, and I leave this behind for you to promote that continuity as new members come in. So I think starting there is good. There's been a number of comments made about data and software resources. And so yes, you can host your open source data sets on OpenNeuro. You can upload your maps in NeuroVault. We like to have a lot of journals are asking now for that data availability statement and making sure you make your maps available via NeuroVault. If you're doing a meta-analysis, also putting the meta-analysis maps on Anima. But just a couple of resources here that we have found to be particularly useful for us that have been addressed in other talks. And again, GitHub is a primary focus of so much of what we've been doing this week, and so I wanted to have us make sure that we were pointing to that. We've had a recent interaction with a large consortium that has developed an analysis pipeline that's closed source, and it's been very we've had to say that their pipeline is lovely, but we really would prefer to use our pipeline that we've developed because it will be openly available on GitHub. And this just promotes reproducibility and reusability. And so this is something potentially you could have a conversation with your PI about as it relates to any particular application. So things that you could be doing, again, the fMRI prep, MRI QC, NiPi and GitHub, then sharing your results. This can mean different things. Maybe not posting your data on OpenNeuro, but maybe sharing your maps to NeuroVault, maybe sharing your figures on Figshare, and making sure that there are as many different opportunities for you to kind of take the little flow chart there and find ways of sharing your information. But one thing that we haven't quite talked so much about is the openness of our analyses and our manuscripts. And so whether or not we can do a registered report might be challenging given your short timeline of how long you're in a given lab, because you have to go through that submission process that may take a little bit longer because you want to get your stage 1 manuscript before you actually collect the data. But pre-registering your analysis plan is something that I ask of all of my trainees. And that's where you sit down, you write out what your hypotheses are, you write out your specific analyses that you will do with that, and then put it on OSF, put it somewhere where you can make it private. You don't have to release it publicly, but it really forces you to actually sit down and make sure that you have all the nuances of your analysis laid out beforehand. And that's not necessarily so much about reproducibility, but about encouraging you to really think about what your hypotheses are and really think about getting your head on straight before you start analyzing the data. Because once you analyze the data, you're troubleshooting this and you're fixing the code for that, and you get overwhelmed by the volume of the results that are coming back, and you're trying to figure out what's my story. And like the publication process, you always underestimate how long it takes to kind of get through a paper. So sitting down and doing a pre-registration where you kind of get as much of that on the front end as possible, it's really, really helpful. And like I said, you can make your analysis plan pre-registered in a private way so that you don't have to get scooped, right? Okay, and then of course we all love our preprints. So other study materials that you can post openly are your figures, your posters, your talks, and there's different venues for you to do that, and my slides are going to be made available, so I will let you grab that later. Before we go, I do want to have sort of a different little bit of a talk about academia and about how hierarchical it is. And because it's hierarchical, there are power differentials, and anytime you have power differentials, you end up with circumstances where it's very easy for individuals to abuse the power that they have. And so this is true regardless, this is not a conversation that's limited to gender identity or sexual identity or cultural or ethnicity. This is something that power begets some troubling situations. And so as as you move through your career, you may experience some circumstances, regardless of who you are, where you are put in some sticky situations, some unpleasant situations, and may have some concerns about those power differentials. So I kind of wanted to give some advice about that as I was closing here, and I started thinking about all the advice that I had been given, and all of it is pretty terrible. It can be very well intentioned, but you hear things like, tell someone, report the abuse, and that's great, but lots of times we have to worry about retaliation. We have to worry about whether or not it's career suicide to say, talk about some of these things. And also you would be horrified to know how many times that I did tell someone and nothing was done, you know? So there are other things that you can hear of, it'll get better, don't worry, I survived, so can you. And that's all meant to provide moral support, but you're just telling me to deal with it. That's also not okay. And so then the other one is like, find a community of support, right? And that's true. You should find a community of support. But what I don't like about this is you're flipping it and you're making it about it's the victim's responsibility to find a way to fix, cope, or manage the situation. And so that's not okay. And so, you know, this is not the best advice, and so I was trying to figure out, well, what is the good advice? And the good advice is that a toxic climate hurts everyone. And so it is our collective responsibility to not tolerate these sorts of things. And so whether or not you have been in a circumstance where you have been on the other end of this, the idea, this is one of my favorite pictures, I'm sorry, because there's no excuse for creating situations that are putting other people at risk. What you can do as a community is that if someone is speaking out, you listen, you do not interrupt, you prevent others from interrupting, and you also, as someone who perhaps may not have experienced these things, seek out their perspectives, right? Go ahead and figure out if there are things that you could be learning about the community that you have not experienced. Twitter is absolutely a cesspool, a hell site. There's all kinds of craziness on there, but go find voices that you can learn from. If you are not a member of a marginalized community, then go follow LGBTQ scientists, follow Black, Latinx, Muslim scientists, all scholars and activists that are doing things that are speaking out. If they're doing the emotional labor, then it's a little bit that you could do to go ahead and listen. And so similarly, change your surroundings and situate your other stuff as an other. And so this is kind of a big thing for me, is that we're not inherently bad people. Well, maybe some people are, but we are not inherently bad people, right? But we tend to gravitate like-minded people towards like-minded people, and we sort of self-homogenize ourselves. And so look around at your group and you're kind of seeing faces that are similar to yourselves, then stretch yourself out a little bit and go speak to other people. And this is kind of a very big point for me, because I firmly believe that a diverse environment is a breeding ground for rich and innovative ideas. You have people coming from different perspectives with different backgrounds. Your science is going to be more diverse as a result of that. And so with that said, I will have a little bit of a shout out to my own university. We are the 4th largest university in the US, and we have a very diverse student body. And I have found it to be a really rich environment to be in. So as you are on the job market, in the US, there are minority-serving institutions, there are Hispanic institutions, there are historically Black colleges. All of these institutions are designated this formally by the federal government based on the population that they serve and meeting criteria for certain percentages. Please take a look at these institutions as you're on the job market, because you will find that there are pros and cons to any particular job, but I have served as a faculty member at 2 different minority-serving institutions, and I have found the environment to be really, really rewarding at both of them. And so with that, I will end thanking my collaborators and my funding support, and thank you for listening. Yes.
I was impressed by how much your results seem to aligned with resting state network, as you emphasized, discussed, and I'm surprised that this is not leveraged in the consensus analysis. There's a lot of voxel-based methods, and you create sort of like probability distribution around them, so you can recreate some synthetic maps. Don't you use directly the resistant network to do some counts and somewhat like align findings across study, if naturally the task-based according for using the distributions?
Okay, say again what you're saying.
Could you do some resting state-based statistics for the meta-analysis?
Oh, sure.
And are there some packages doing that?
No. Not that I know of. Katie?
Direct statistics on resistant networks to the voxel-based, if you naturally the results following this pattern then.
I don't think any exists.
I don't think so either.
I don't think there's anything that's specifically toward either resting or network.
Yeah.
There's been some work by Roberto on actually looking at the meta-analysis of connections. That's something that in the earlier version of Brainmap, there was a way of actually looking at how many paper reports, how many co-activation do you get, and therefore how many functional utility things, and what will be the meta-analysis on those things. I think there's still some things, and I have actually a student working also with Jerome in that direction as well.
I think a number of the image-based techniques could be readily applied to functional things. So things
Right.
like just activation maps.
And Bharat did some stuff where he was looking at using Brainmap data and doing some graph theory stuff, but I think that was just a couple of pubs, not necessarily any tools that were being disseminated. All right, guys, thank you.